Okay. Um, so good morning again, and this is lecture 1, and the lecture will be posted on Blackboard and YouTube. And so hopefully you will subscribe, and then you will see the lecture on YouTube. And I will remove the old lecture so to avoid any confusion. Okay, so in today's lecture, we'll cover 2 chapters, and those are really fun and easy chapter to read, and you are required to read the entire chapter 1 and chapter 2. This is for today's lecture, and in the future, you may not have to read the entire chapter at the beginning, but I'll explain that later in the future. So we'll discuss 2 chapters, microorganism and microbiology, and we'll have a quick overview of the microbial life. So for the chapter 1, I will introduce you to microbiology and comment on the history of microbiology. So microbiology obviously is a science that studies micro, and anything that's invisible to your naked eye is micro, or any organisms that that have a life cycle that go through a small a period that have a tiny a cell that requires microscope to view. So some of the parasites will be covered as the microbe and in the lab. Although we have a parasitology, I encourage you to study. One thing that it's interesting is that we want to view the microbes as cell, and I'll discuss a bit more, and we'll look at the natural environment, and we'll look at the microorganism and human. That's it's very important, and especially in the pandemic, right? So microbiology is the study of microorganisms and their impact of on our life, okay, good and bad. And so here are pictures of a purple bacteria, and this is a green or yellowish cyanobacteria. Here's a picture. Does anyone have, so why do bacteria have color? It's always fun to think about. Why some bacteria are purple and this bacteria is yellow and actually green? Can you repeat your?
Said, is it because of what they eat or whatever they're surrounded by?
Okay. Yes, they depend on the color, and that's they eat, and there's a curriculum, a general public would say bacteria eat. We'll get to that. Well, for scientists, that's how they get energy, or they need something. Okay, so these are both of photosynthesis bacteria, and so they need these pigment to harvest light. We'll come back to that later. Okay, so along the way, I wanted to drive home the theme that the microbes are always living in a energy deprived environment, energy poor environment. Okay, so they're very efficient with energy. So they only use energy for things they are absolutely needed. Okay, and we start to turn off the light when you go away, and they do that long time ago. Right? If there's a process they don't need, they shut it off immediately. We'll get to that. So when we study microorganisms, we're really looking at them as an individual cell, individual living organism. And for anything living, one of the things they do, they have to get energy from somewhere. So they consume food. Okay, like those two other bacteria, well, they get energy from light. Right? And most other microbes get energy from, say, carbohydrate, right? Or think about the microbes that you have in your digestive tract. What do they do? Where do they get food? So they get food from whatever you eat as a breakfast. So did you get bread this morning or bagel this morning? Yes, yes, Maria. Thank you. That's right. So I hate to break up to you. This morning you had breakfast. Yes, you had breakfast. But what did you do? Well, you got food and you fed the microbes in your digestive tract. That's what you did. Okay, I'm just kidding. You do get food, and you, your small intestinal tract is involved in taking up all the food, the carbohydrate, sugars. Okay, microbes reproduce, and if you are making notes, which I assume I structured my my slide so that you can make notes. So the notes you will make is to put 3 stars or however emphasis you want to make. This is the ultimate goal. Even though we treat microorganisms as individual, they don't have a brain, right? So what's their goal in life? Reproduction. Reproduction. That's it. Their goal, the goal of microbes is to reproduce. Anything the first thing you want to think about, why does microbe do this? You want to think about why they reproduce. That does it help from reproduction? So, for example, we now deal with the Delta variant, right? The Delta variant become more infectious. Is it good for the virus, or is it more important for being deadly? Right? Initially, the virus was very deadly, but now it become much more easily spread. Well, there are 2 routes for reproducing. They can go into your body and reproducing deep in your lung, and eventually kill the people, right? Because that's. But then they can also reproduce really fast in your upper respiratory tract, and where you sneeze out and they go to the next person, and then go to the next person. So if you think about now we call it SARS-2, right? COVID-19 is SARS-2, but SARS-1 was really deadly. It's coronavirus, right? But the spread was very well contained because they were infecting the person. They didn't go spread. And so now the SARS-1 no longer exists, and SARS-2 is spreading all around the globe. And so so yes, so the way you view this is think about what's better for the virus to reproduce. Now, obviously, they don't think we can trying to analyze, better understand, and then predict, because you can almost predict the virus epidemic always goes from very, very deadly to very, very easily spread and less deadly. And then at some point, yes, we're going to all have it. We've all seen it. We all essentially naturally vaccinated. But they still reproduce somewhere in our population, just like the flu, right? Flu comes back every single year. Yes, like no, not the, yes, common cold, and they don't kill you, they just make you sick, but the common cold never goes away. All right, so this is the emphasis. All right, the next I wanted to explain differentiation. The differentiation for microbe organism is that when they some of the microorganisms can differentiate. This is a verb, can change into, and this is an irreversible change, and can change into a different environment. They produce different set of genes and produce. So like if you fly to South Pole or North Pole now, what do? You put on more heavy jacket, right? But what does microbe do? They can't put a jacket on. They produce a set of genes that allow them to deal with environmental change. Okay, so we have that. And microbes, as we learn more and more about it, microbes communicate with the environment. They can sense the environmental change, and they can communicate with hosts, and they can trick hosts into open gates so that they can infect you, and then they communicate amongst themselves. Okay, and so for example, just like you walk into, if I blindfold you and make you go to an elevator, would you be able to tell the elevator is filled with people or with no one? You would say, I can smell it. The room elevator is filled with people, especially there's one person just came out of the gym, right? Okay, so that's a chemical signal. We'll talk about that. And they move around, and then they evolve. And we just talked about how viruses evolve, right? They change. They evolve. The reason they evolve fast is because their lifetime, the generation time, is very short. Our generation has come to us, we count our generation as like 20 years a generation, right? That for bacteria, for E. coli, that's your model organism, it takes about 30 minutes in the best condition to reproduce from one to another, to two, from one to two. Okay. So those are the highlights that we want to think about when we treat microbe as individual. Those are important. And we need to understand their their environment. Everything had to do with their particular environment. Microbes are adapt into a particular community and environment. The question about what's the differentiation between reproduction and differentiation. Differentiation is at the individual level. They will produce a set of new genes that they go into a environment. Reproduction is go from 1 to 2 to 4. Okay, hope that's clear. And so we need to understand their environment, and the microbes all specialized, and they all dependent on each other, and they as we will learn, they help each other. And some of the microbes are in our environment as well, right? We depend on microbes for some of the things, which I will talk about. And here, just a picture of a sewage sludge. Okay. Now, when I say sewage, the image isn't very good. I hope this image will change. So from this point on, I know you've never seen a sewage was so pretty, right? And say, or maybe you want to save this picture. Next time when someone says sewage and says, ew, and you say, no, 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 sewage is beautiful. So, well, not beautiful, but this sewages are microbes stained or labeled. This word stained really means label. Labeled with different dyes and specialized in binding to different bacteria. So you will see different bacteria being labeled in this case. So as we know now, we already know sewage is a mixture of hundreds of different bacteria. And this picture really brings it to you and to say, okay, yeah, this is a mixture. So now I wanted to talk about microorganism and their relationship with us. Well, the first thing you think about is disease. We're in a pandemic, and we worry for the virus. We also worried about the loss of effective antibiotics. So we worried about bacteria infection that will come up. And they and so wait. Sorry. So this is in the 1900s. That's where the people, where the microbes, bacteria, diseases are more prevalent, kill more deadly back then. And so see tuberculosis are still there, and influenza, pneumonia, and the heart disease and stroke, kidneys were there, and cancer, but they don't. They're not the leading cause. But now with the antibiotic, microbial infection is no longer listed here. Heart disease and cancer are the leading cause of the death. HIV is still here, influenza is still here. Now these are flu, not COVID-19. Well, it's also coronavirus. So that's the disease, but microbes to us are not just the disease. We rely them for food. Any food that you can think of depend on the microbe.
Yeah, yogurt.
Yeah.
Yogurt, yes, that's the healthy. And the yogurt is interesting. It has living organisms in them, right? You know that, right? And pickles, cucumbers. And if you have, yeah, kimchi, yes, yes. Cheese, exactly, yes. Those are all processed by microbes. Bread, no one drinks beers and wines. Oh, tasty beers and
Is it like
wines.
kombucha as well?
Yes, yes. But have you ever thought about the which we'll get to it in the future? Well, we know beer and wine contains ethanol, right? is why do bacteria produce ethanol at all? Just for our consumption?
It's a byproduct of fermentation, right?
Yeah, byproduct is the nicer way of saying it. It's a waste product. The yeast takes sugar and ferment it and get the energy, and then ethanol is their waste. I'm sorry, I hope I didn't ruin your food, but you know, that's happening in the environment. So this is a vivid example of how one microbe is producing a waste product. The next organism, us, is taking it on as happily. We'll get to that. And the microbes heavily involved in agriculture. Nitrogen fixation is one of things that we study. So how do bacteria help plants like beans and soybeans and all those legume families fix nitrogen from nitrogen air gas into ammonia. It's an energy consuming process. Or you go to Home Depot, buy the fertilizer, right? But that fertilizer is really terrible for the environment because it's chemical. Eventually will destroy the soil particle, which we'll come back to this later. And it's always involved in energy and then changing the environment. Energy, like meaning the ethanol production. The gasoline has 10% ethanol, right? And biotechnology, anything you think about, it's all from micro. Our understanding of molecular biology, all the current pharmaceutical, all came from studies of bacteria or virus. And the first cloning vector was a virus called M13. It's a linear virus. Later on, I'll talk about that. All right. To the current treatment of the vaccine, right? The mRNA vaccine that we all took. So those all came from studies of micro. All right. So now I wanted to comment a little bit, highlights on the history. History of microbiology compared to other science is less, shorter. But shorter doesn't mean it's less significant. So let me go through, highlight some of the people history in the history. I'm expecting you to read in the textbook. All right. Now the textbook is free, only one click away. So there is no excuse or to come back and says I don't have the textbook. All right. So I expect everyone to read chapter 1 and chapter 2. Leeuwenhoek, obviously, being a credit you've read many times in many different versions of textbook. And here is the replica of a microscope that he used to see this cells, red blood cells. This is view in this microscope. I'm sure you've seen this picture showing you before. Does anyone know how this thing works and what is the problem this is solve? When you think about microscope, you think about the microscope in your teaching lab. But have you thought about this microscope actually able to see red blood cell? Okay, so how does this work? Well, can everyone, when I show slides, can you all see me talking? Not so certain because I'm.
Yes. You're in the bottom corner.
Okay. Oh, that's good. Okay. So can everyone see? Well, I see a tiny 1-inch picture of myself at the bottom. So I can't really say. So I'm holding a piece of paper, right? And think this, oh, if I click.
If we click on you, we can see you. And if we click on the slide, we can see the slide. So we can go back and forth.
Oh, good, good. Okay, so can you see me for now for a second? Okay, because I want to show you a piece of paper, and now it has a hole. Imagine this hole has a magnifying glass, and now you've all know magnifying glass, right? If you hold a magnifying glass and you're trying to magnify, and what's happening is then your hand is shaking, right? And so it's going to be hard. Or another way to think about this is binocular, or right. So things are moving, and when you use magnifying glass, the more you're magnifying to this level, any slight movement, any tiny movement will make the image blurry, right? So that's the limitation of your magnifying, using magnifying glass to see things. So then what he did is that he put a needle. Can you see I'm using my hand as a needle? And he puts the needle to this plate, the copper plate. And then he put the sample on the tip of the needle. So what does this solve? Well, there is no relative movement between the sample and the magnifying glass. So that's fixed. You see, it's even adjustable. So now you can see why he was able to see this microscope, see red blood cells using this so primitive looking magnifying or microscope. Yes, so the magnification, okay, let me go back. The magnification, the power of magnification is dependent on the curvature of the lens. The flat lens have no magnification. So if you think about your microscope, if you next time when you go back in the lab and take a look under the objective lens, the 4x is giant piece of glass, and then if you look at the 100x, it's a tiny pinhole. Why? Because the curvature. The higher the curve, or the more curve, the more magnification you get. That's why the lens looks small, but the magnification power is greater. Does it work? Okay. So that's why this is important. This is how this microscope works. Okay, that's a giant step forward. Then once we figure that out, and then they use another. So what you essentially do is you have, this is the objective lens, but it's so tiny, the image is so tiny, you have to put your eyes to it. And you put another magnifying glass here. You put another switch my hand. You put another magnifying glass up here to magnify the image from this lens, and so now you have compound microscope. Okay, good. Okay, so the next, so that's about the microscope, and the next giant in microbiology is Louis Pasteur. And you heard about milk needs to pasteurize, right? And heating at 65 degrees kills all microbes. But this is all, but his major contribution, even bigger contribution, was to prove life can only come from life. And this is his experiment. Because back then, in his time, people just see soup puts into, if you leave a nice meat soup or fish soup overnight on the counter, the next day it smells terrible. And if you leave it longer, you can even see microbes in there, right? So people at that time think non-life, life can come from non-life. And soup has no life, but if you leave overnight, living things will come out. And he set out to prove, no, life comes from life. This is the experiment he did. So he put soup in a flask and he twisted, heat the glass and twist it. I know you guys don't do that in the chemistry lab nowadays, but if you get a chance, bending glass is really fun. Okay. The Bunsen burner, you heat it up and the glass becomes soft, and then you can just twist it. And so he twisted the glass and then he boiled it. Sterilize it. And then let the liquid cool down slowly. And so now the microbes in the air will come and get trapped here. Okay, because it has to be slowly, because if it goes too fast, the air will go quickly, carry the microbes over and into the soup. And so this is open-ended, and the liquid remains sterile for many years. Right? Now, so this is his experiment to show that microbes from the air can spoil the soup. And so this he did even further. So if you tip the flask over, you can actually, and tip it back, the soup will get spoiled. Okay. So that's his experiment to show that life can only come from life. Right. So the next giant that I like to mention is Ferdinand Cohen. And he was the first, he was trained as a plant biologist. Then he got interested in bacteria, especially the bacteria that forms endospore. And Bacillus is the bacteria he studied. So he's considered to be the person who started bacteriology. Now, another branch that studies bacteria. And then Robert Koch, and this is Koch's postulates, which is the rule that people follow to prove a organism is the causing a disease. So now, so for example, this is his steps described, and so organism must be present in all the diseases, but absent in the healthy animal. Then you have to isolate, and then you have to get grown in a pure culture. So I would like you to highlight these two words, pure culture. In this, for microbiologists, pure culture equals a population of identical bacteria, identical cell, meaning pure. Okay? Pure culture is a population of identical microbes. And so the cell then from this pure culture can cause the disease. In healthy animal, and then you have to re-isolate the bacteria from the healthy animal to show it's the same as the original. So these are the steps that one must go through in order to demonstrate that this is the organism cause a disease, the linking a disease and an organism. So the next person is Richard Petrie. I'm going to let you guess why. What is his contribution? Very nice. 3 of you says Petri dish, the Petri dish. But you will all, none of you will get the point for saying the Petri dish. And guess why? Look at the text. How did you type? Ah, yes. Thank you. Petrie is a person. From this point forward, if you type Petrie with a lowercase p, then you just failed. Okay, so that's a few things that you can tell whether a person is really a microbiologist or bacteriologist. Is whether how they spell Petrie. All right. The next word that you need to spell correctly is Grandstein, which we'll get to it. Now, microbiology is a living science, and we're still moving, and there were so many to discover. And in 2000, Edward D. Long was credited to discover the Archaea as a main branch of the microbe. Okay, there are 3 domains of microbe, and he is one of. He established Archaea as one of them. All right. So those are the general concepts of microbes and microorganisms. So I'm going to pause for just a minute or so, so I can see if you have any questions. Any particular issue that you find really fascinating, interesting, that we can follow up in discussion in future?
I do have a question, Professor.
Yes, go ahead.
It was regarding the Pasteur, and the the pipette. It was a pipette, right? Or no, it wasn't a pipette. It was, what is it called? Was it the Bunsen thing?
Well, Bunsen burner, yes.
Yes. So when so was that to show that life can only come from life, or life can come from not living? Like
No, it's just proof, right? So basically, the notion at the time was life can come from non-life, and by doing this experiment, he shows life cannot come from non-life, because if you prevent things from going into it, if you isolated the soup will remain as a soup, not get spoiled. Nothing will get in, right? And so he further demonstrated whatever got soup spoiled is the thing that probably comes from the air, right? At the time, that then he can crack open the flask and see what's in there. They can see the microbe floating around and swimming around.
okay, thank you.
Yes. Okay. That's our short break, and then we'll go back into lecturing. Any other questions? I have 21. All of you didn't fall asleep, right? Because last semester, my daughter is next door, and she thinks that my lecture is boring. I worry that I'm putting my student into sleep. So, okay. Okay, so we're going to go back and start discussing the second half. Okay, hold on. Okay, so the for the next 20 or so minutes, I'll give you a very brief, quick overview of of microbes, and so what are the things we're going to be talking about, and this should be viewed as a review. And everything I will be talking about in the next 20 minutes is something you're supposed to know and know very well. All right. And this is almost like a checklist. Okay. So a prokaryotic cell. This is a bacterial cell, has cell wall, membrane, and ribosome. The only structure that's somewhat visible is the nucleoids and the size. And the size is important. And at this point, I wanted to the size of E. coli. E. coli will be the organism that at the end of the class, you'll be able to say, my best friend is E. coli. I can tell you all about it. Where does it live? How big it is? What does it eat? What does it like? How does it move around? Okay. Okay. E. coli. The size of E. coli is 1 micrometer by 3. So this is a half micron or half micrometer. And so an E. coli is slightly shorter than this. It's 1 by 3 micrometer. It's extremely important if you put a unit on it. If it shows on the exam and you didn't put a unit, it doesn't count. Right? And then, so this is a picture of eukaryotic cell. It's a lot bigger. And how big? Relative scale. This is mitochondria. Mitochondria is originally, we all believe, from a prokaryote or bacteria cell. It was captured by this cell, and then the specialized in energy production. So this is mitochondria, and this is chloroplast. This obviously is an ideal cell, not a real. And so this is a transmission electron microscope of a bacteria cell, right? Nothing really pokes out structure-wise, but this has some things inside. We'll talk about that later. This is a picture of an eukaryotic cell. So you can see the nucleus and you can see the mitochondria, right? Has these membrane. And nucleus versus nuclear, and that, okay? So nuclear is the area that was stained. And if you zoom in, you can see the DNA. Okay, there was so much DNA winding around, right? And if you, this is prokaryote, but if you see eukaryotes, you can see mitosis, and these are the chromosome that's stained. But if you were to use a microscope and zoom and looking at your skin cell, looking at your hand, and zooming, hypothetically, will you be able to see chromosome? Would you see chromosome? Why not? Catherine, you want to take a shot? Why? If you zoom in, you said you won't see chromosome. Why not? Do we have skin cell has chromosomes, right? Now, none of your answer is correct. I'm reading your text. Anyone? What's mitosis? Is your skin cell going through mitosis? Aha. You forgot that part. The cells only you will only see chromosome when cell go through mitosis. Most of the time, the DNA is relaxed in the nucleus and within the nucleus, and so you zoom in, you won't see it. They only form chromosome when they divide and then when they try to separate. Okay, so that's a sort of tricky question that I will ask. Okay, you better be on your toe. So that's something that you're supposed to know from your genetics and intro bio. So for bacteria, for eukaryote, for us, we have 2 copies of chromosome, but bacteria only have 1 copy of gene, 1 copy. And so that makes it easier for us to study, because if you're not, you disrupt 1 gene, you can see the phenotype. Versus humans or eukaryote organism, you don't. You have to get into homozygous, right? In terms of size, they're very different. So here's virus, here's prokaryotes, here's the nucleus, and together there is a eukaryotic cell. Okay, so that gives you some sense. So now I want to talk about the virus, small things. The virus, here is an example of Rapnel virus. And this looks like a bullet. And if you heard about rabies, and that's one of those viruses, rabies virus looks like this. Okay, and they tend to replicate in nerve cells. And so go from nerve cells and infect the next nerve cells. And so when you have a possible bite of an animal with rabies, and you want to see a doctor really quickly because there's no cure for this viral infection. The only way you can capture it is get a vaccine immediately, so your body can stop these virus from replicating. And it's trying not to get your face bitten by a animal that has rabies. Not get bitten, but because it's the rabies virus travel to your brain, it's where they can kill you. If it's closer to the brain, like if it's tip of your toe, then because they go replicating one cell, then go to the next, so then you can have enough time for your body to react. Right? We know at least 2-3 weeks, your body able to produce vaccine antibody to stop the virus. But if it's unfortunate, your face gets bitten where the virus starts, and then you have shorter amount of time. So that's an animal virus, and this is a bacterial virus called Lambda virus, and that's what they look like. And the obviously this is the COVID-19 virus. we will talk about that at the end of the lecture, no, close to the end of the semester. We'll go through, we'll be able to understand different cycles of microorganisms. We'll be able to finally understand what each of things are, okay? So to understand how, what we're doing, what monoclonal antibody, like Regeneron is making monoclonal antibody as a treatment to bind the spike protein, and why spike protein is so prominent. So all those questions we'll learn throughout the semester how microbe works, and we'll be able to apply. So those are general overview of the microbes, and I wanted to go into specific microbes. So the microbes, organisms, when we think about it, is part of living organism on the earth. Everything living on earth can be separated into eukaryota, archaea, and bacteria. This is the 3 domains structure. This is all based on the ribosome. All living organisms have ribosome, and the ribosomes are different, and we'll talk about that later. This is how they can be separated. And all living organisms have to get energy, and we can separate them based on how they get energy. So you can have energy, a microbe gets energy from chemicals or light, and so there are chemotrophs or phototrophs. Within the chemotrophs, you have how they get energy from organics or inorganics. And so now you have chemoorganotrophs and chemolithotrophs. And so those are different types. Okay, and so you can separate them how they get energy, and then they can we can classify them based on how they tolerate extreme environment. Okay, so you can name them based on what they like: low pH, high temperature, and they all have different names. High salt. Okay, so these are extremes of extreme files. So in terms of bacteria, they're part of prokaryotes. And the prokaryotes can be further grouped into these groups, which next time we'll talk about in detail. Can be grouped into what's called Proteobacteria, gram-positive, and all those different groups. Throughout the semester, we'll have a chance to talk about each of them. And so first, I'll give you examples of Proteobacteria. Proteo meaning bacteria that have multiple shapes. And these are the bacteria that the earlier microbiologists first saw. So they say, the bacteria could be in multiple shapes. So they then create a term called proteo, meaning multiple shapes. Only later on they learn that different shapes of bacteria represent different species of bacteria. And each bacteria maintains one particular shape and the size. So this is an example of a sulfur oxidizing bacteria. And this is examples of gram-positive bacteria, Bacillus, Streptococcus, and those are common on your skin. Streptococcus is common in your throat. When you have strep throat, this is the microbe. And this is cyanobacteria. They're green. They tend to live in water. They do photosynthesis. And so we just talked about proteo, gram-positive, cyanobacteria. There are other microbes like planktomyces. Here are examples of planktomyces, more like fungi, right? They have, and then you have green bacteria. Here's an example of green sulfur bacteria. They literally are green because they're doing photosynthesis. And sulfur is that they actually produce sulfur. Is the end product, but they use hydrogen sulfide as the energy, the donor of electron. So these are the details we'll talk about. Here is a spirochete, and that's the one. This is the one that causes Lyme disease. I'm flashing through. Don't get too nervous. This is the level of detail that we require for this particular lecture. The next is Deinococcus radiodurans. Based on the name, you would guess this is a radiation-resistant bacteria. All of us are sensitive. All living organisms are very sensitive to radiation, except this bacteria. For some reason, it's very resistant to radiation, high resistance. Okay, and then you have other green sulfur bacteria. This is living in a chain. So now we're down to, and then you have some, so those are examples of bacteria. Archaea is a group of bacteria live in extreme environment, either very salty, like halo, or thermoplasma, or very, very hot. So they tend to live in very extreme environment, and it's difficult to grow them, and it's difficult to get them, create a condition to grow them. So so we know a lot less about these microbes. They have very strange shapes. And we'll talk about later because their cell wall molecule is different. So they don't have regular shaped. So E. coli has one, for example, is rod-shaped, and they tend to be one particular shape. Right? But Archaea, even within a species, they could be very different. All right. So, and eukaryotes. There are actually a lot of, we're part of eukaryote, right, domain, but there are actually single cell eukaryotes. And some of them you already know, like yeast is one. And then you have other flagellates and algae and green algae. So here are some examples that you will see. So here's green algae, and this whole thing is green algae, and it's enclosed in a membrane with chloroplast, and chloroplast is where they capture energy from the sunlight by photosynthesis and provides the energy for the green algae to grow. Now the membranes are fragile, and so something die, the chloroplast is floating outside. Okay, and so here is a typical mold. These are the spores. We'll talk about that. And so here is something called the protozoa paramecium, and in the next few weeks we should be able to provide samples. They live in natural environment, and they are very fun to watch. They can swim around. And if you look carefully, the microbes are all around us. Okay, and so and here's one example. Is it's a nice story. You can see this is a dinoflagellate from Bronx River, Bronx River. And you think Bronx River is a toxic dump, right? Nothing growing. No, actually, if you walk by Bronx River, it's actually much more restored. Um, Dr. Joe Racklin is part of the effort to restore Bronx River and restock with fish. So you will be able to see someone's fishing in there. Although I'm not sure it's the fish is edible from there. So one day he came back from Bronx River and says, "Hey, microbiologists, I saw something rusty-ish floating in the water. I'm wondering, is that really rust or something growing?" So here's a bottle of water. Can you tell me what that is? It's like, I'm a bacteriologist. I don't know these things, but let me take a look under the microscope. So I put it under the microscope, and this is what I see. And I was like, what is it? Luckily, I have a book, dinoflagellate. So I went to the book and found the closest one protozoa that's like what I've seen. This Professor John Lee, anyone from City College in my class? Occasionally I have a few because they come to take my microbiology class. And he's a professor there. He wrote this book and he was an expert in this. So I have his book, I cheated, and I say, well, I told Dr. Racklin, well, this must be Gynodidum sanguineum. I know I pronounced it wrong for sure. I'm gonna have to. So I just handed him this picture, and we had a lot of fun. Say, okay, fine. He accepted that. And as we were watching, and this is happening, anyone want to know why this is happening? I'm saying the demise of dinoflagellate from the Bronx River. Anyone want to guess what's happening? Yeah, it's budding. And why does it bud? Why does it do this? Let me give you some clue. Reproduce. I was looking under the microscope, and I put a drop of liquid from Bronx River onto the microscope. I put a cover slip on it and look into the microscope. So as time goes on, hypotonic. Well, no, yes, similar, but from heat of the light, no. No, it's think about water, and what's happening to the water in on the slide when you keep it on the slide for a long time. If you put a drop of water on a glass slide, the glass slide will, the drop of water will what? Will the drop stay there forever? Evaporate. Excellent. What happens when the water starts evaporate and the distance between the cover slip and the glass slides gets what? Smaller and smaller, narrower and narrower. So I'm very, very sorry to, desiccation, close. And so I'm very, very sorry to this dino. Yes, excellent, Sean. It's the cover slip is pressing down and being, yes, sorry, yes, I'm very crude. It's squashed, and that's why they died. Okay, so So this is these are this is a quick overview of all the microbes that the eukaryotic, prokaryotic around us. And I hope you enjoyed it. And this is really an overview. And we'll go over this in detail.